Alright, so previously we have looked at the structure and function of amino acids, and now we are going to continue with the next topic, which is about proteins. Amino acids are the monomers of proteins. Ribosome is the organelle of the cell which synthesizes proteins. This is a diagrammatic representation which shows a ribosome here. Ribosome is important for the process of protein synthesis. It binds to the messenger RNA, shown in red color here. The messenger RNA contains codons. The transfer RNA molecules having complementary anticodons on them, they can come and bind to the ribosome along with their amino acids. And the amino acid, it carries out the process of peptide bond formation here. So in this way, amino acids one by one are going to be added to produce a given protein. In this way, proteins are synthesized, and then the proteins, they would fold into a particular structure as they're being synthesized. This is again a depiction of the various levels of organization in protein. The first level of protein structure organization is called as the primary structure. The primary structure is essentially the sequence of amino acids in a given protein, that in what order the amino acids are attached to one another through peptide bonds. So that is called as the primary structure of protein. In this session, we will talk more about the primary section of structure of proteins. The amino acids of proteins, we have seen their chemical natures during the previous session. The amino acids interact with one another through different types of non-covalent interactions as well. These non-covalent interactions include van der Waals interactions, hydrophobic interaction, hydrogen bonds, ionic bonds, and so on. So in this way, through different types of non-covalent bonds, the different parts of the protein, these amino acids, they interact with one another and they fold into local structures. These local structures are called as the secondary structure. One secondary structure, which is called as the alpha helix, is represented in this picture. Now, many regions of the protein, they interact with one another and they give rise to a global conformation of the protein. This is called as the quaternary structure of the protein. This is called as the tertiary structure of the protein. And then for those proteins which have only one polypeptide chain in them, their highest level of organization is tertiary structure. But all proteins are not made up of one single polypeptide chain. Many proteins, they contain more than one polypeptide chains as described in this figure. So each of these polypeptide chain has its own tertiary structure, and they all interact with one another to give rise to this entire multimalic protein. So those proteins which have more than one subunits in them, their structure is called as quaternary structure. So these are the four important levels of protein organization. This is what we have seen previously as well, that amino acids, they interact with one another through this peptide bond. So what is happening on the ribosome is that the amino acids are brought by the transfer RNA molecules. These transfer RNA molecules, they deliver their amino acids to the ribosomes. And the ribosome, what it does is that it catalyzes the formation of peptide bond between the carboxyl group of one amino acid and the amino group of an incoming amino acid, thereby forming a peptide bond. We've talked about this earlier as well, that the peptide bond, it is a condensation product between carboxyl and amino. So the chemical nature of the peptide bond is that it is an amide linkage. In this way, now the carboxyl group of this amino acid is free, and the next incoming amino acid, its amino, can react with this free carboxyl. In this way, the process would continue. This is a diagrammatic representation of a peptide. And in this peptide, you can see that this is the first amino acid, which is alanine, represented here. So this is the amino group of alanine. And this would be the carboxyl group of alanine. This is the side chain, the alpha carbon. So till here, till this carboxyl, that's the amino acid alanine. Now the carboxyl of amino acid alanine would undergo condensation with the amino group of threonine. So this blue atom here is the nitrogen. So the carboxyl, it forms a peptide bond with this amino. This is the alpha carbon of threonine, This is the carboxyl group of threonine. Now the carboxyl group of threonine will undergo a condensation to form a peptide bond with the amino group of glycine. In this way, the amino acids, they will undergo this process of peptide bond formation one by one, and eventually at the last amino acid you can see that the carboxyl group is free here. So the two ends of the protein, they have chemically distinct groups on them. One end of the protein contains a free amino group. This is called as the N-terminus of the protein. Sometimes it is also called as the amino terminus. The other end of the protein, it contains a free carboxyl group on it. So this is called as the C-terminus of the protein, or sometimes called as the carboxy terminus of the protein. Every protein or every peptide, it contains the N-terminal free amino group and a C-terminal free carboxyl group. So the two ends of the protein or a polypeptide chain, they have chemically distinct groups on them. When we write the sequence of a protein, that is the order of the amino acids, we start from the N-terminus, and then we start writing the sequence of the protein towards the C-terminus. As depicted here for this peptide, alanine is the first amino acid, so this is the amino acid which is present on the N-terminus. It contains a free amino group on it. The next one is threonine, glycine, alanine, and so on, and eventually the last amino acid on this polypeptide uh, is glycine. Glycine is on the C-terminus, and it would be containing a free carboxyl group. So when we write the sequence, the primary sequence of proteins or peptides, so we start from the N-terminus and eventually move to the C-terminus. This is the primary structure of insulin. This is bovine insulin. So the insulin molecule, it contains two chains. This is called as chain A, the other one is chain B. So this is the N-terminus of chain A, and this is the C-terminus of chain A. Chain A contains 21 amino acids in it. Chain B contains 30 amino acids, and this is the N-terminus of chain B, and this is the C-terminus of chain B. We talked about disulfide bonds previously as well while discussing amino acids. So disulfide bonds are formed between the sulfhydryl groups of cysteines. These cysteines, although you can see that they are away from each other, they contain several amino acids in between, but the structure of the protein is such that these cysteines, when the protein folds into its proper three-dimensional geometry, these cysteines are very close in space to each other, and therefore under oxidizing condition they can form a disulfide bond. Similarly, a disulfide bond is formed between these two cysteines between chain A and chain B. Similarly, between these two cysteines of chain A and chain B. So these cysteines, they lie very close in space, although they look on the primary sequence, they look far apart from each other, but actually when the protein folds into the three-dimensional geometry, these cysteines lie very close to each other in close proximity, and hence their sulfhydryl groups, they can undergo oxidation and form the disulfide bonds. So this is an example of a primary structure of bovine insulin. Now we are going to look at the process of determining the primary structure of proteins. One of the ways which is very widely used these days is the computational method to determine the primary sequence. That is, we obtain the sequence of a gene and then we can deduce the sequence of amino acids. The other method is an experimental method where we take a protein, purify it, and after purification we subject it to a chemical reaction. That is called as Edman degradation, based upon the name of the person who devised this method. So Edman degradation is a process, it's a chemical series of chemical reactions, and through those chemical reactions we can identify which amino acids are present in the protein. So this is an experimental method in which we determine the sequence of amino acids. The computational method works in the following way, that now we have very sophisticated and robust methods of DNA sequencing. So, and we can sequence very large pieces of DNA. We can sequence entire chromosomes as well. So first of all, we carry out sequencing of DNA and assemble those sequences. Then we have computational programs to find the genes in that given sequence of DNA. And after finding those genes, the genetic code is also known to us. So there are computational programs which we can use to determine the amino acid sequence of those genes. That when those genes are going to be used for protein synthesis, what will be the sequence of the amino acids? And this is because we know about the genetic code. Well, that's the same codon table which you have studied earlier in genetics or cell biology. So once the sequence of the codons is known, those codons they could be decoded in form of the respective amino acids. In this way, the primary sequence of the proteins it could be deduced based upon the sequence of the coding DNA. But there could be several anomalies in that. For example, one of the things is that some genes they contain more than one start codon. So protein synthesis may take place from one start codon or the other start codon. So in this way, we cannot exactly determine the sequence of the protein. Moreover, eukaryotic genes they are split. There are these non-coding regions present in the genes which are called as introns. And the introns they are removed and the coding part of the gene which are called as exons they're joined together. The process of joining is called as splicing. Sometimes several variants of the protein arise because of the pattern of splicing, so that how the coding regions of a gene they are joined together, so that can produce variations as well. So although we have this convenient method of computationally deducing the sequence of the protein, but in some cases we need to determine the sequence of a given protein experimentally as well, and that is mediated by the process of Edman degradation. The process of Edman degradation is based upon the following steps. First of all, the protein of interest, it has to be isolated and purified. This method will not work if the protein is not purified because there will be contaminants, there will be unwanted proteins, so they will produce misleading results. So the protein must first be produced in sufficient amount and purified. After purification, the protein, which is a very large molecule, must be broken down into smaller fragments. In other words, into short peptides. Then these peptides, after the breakdown of protein, are separated and purified. Then each of the peptide which is obtained by the breakdown of the protein is subjected to Edman degradation. Edman degradation is a process, it is a series of chemical reactions which is meant for identification of the amino acids present in a given peptide. This process is mediated by this chemical substance, phenyl isothiocyanate. This figure shows the chemical reactions which take place during the Edman degradation. So here you can see that this compound phenyl isothiocyanate. So this is a benzene ring. When benzene forms a single covalent bond by substituting one of its hydrogen, that is called as the phenyl. So this part here you can see that one of the hydrogens is removed and the carbon is forming a single covalent bond with nitrogen. So this part of the molecule is phenyl. The word thio means sulfur. So that means it contains a sulfur. Cyanate. Cyanate means that it contains a cyanate group, CN group here. So this compound is phenyl isothiocyanate, abbreviated as PITC. So when the protein is broken down into its constituent peptides, the peptides are separated and purified, after which they are subjected to the process of Edman degradation. So the first step is the nucleophilic attack of this amino group. This is now the peptide, and this is the amino terminus. At the amino terminus, the amino group is free. So through a nucleophilic attack from this amino onto this carbon, it forms a bond with phenyl isothiocyanate. The molecule undergoes structural rearrangement, and this rearrangement results in breaking the bond between this carboxyl and the amino. Note that this is the peptide bond present between the first amino acid and the second amino acid. The first amino acid is represented in blue color. So this structural rearrangement in the molecule results in breaking the peptide bond. When a peptide bond is broken, the first amino acid, or in other words, the N-terminus amino acid, is now conjugated to phenyl isothiocyanate. And the second amino acid now becomes the N-terminus amino acid of the remaining peptide chain. Now, depending upon this structure is the same for all of the amino acids. That is, all amino acids, they contain alpha carbon, they contain a carboxyl group, they contain an amino group. So this region of the molecule is constant for all of the amino acids. However, every amino acid, we have seen that previously as well, contains a unique side chain, which is represented by R here. So the R group or the side chain is different for each of the amino acids. So the molecular mass of each of the side chain is known. Depending upon the type of the conjugate produced here, so the amino acid which was present on the N-terminus could be identified by comparing it with the standards. So the second amino acid now becomes the N-terminus amino acid. In the second round of Edman degradation, now this amino acid will form a complex with phenyl isothiocyanate and will be removed. So the third amino acid will now become the amino terminus amino acid here. So in this way, through the series of these steps, the amino acids are removed from a peptide one by one from the amino terminus, and based upon the standards, the type of that amino acid is determined, that which amino acid it is. So therefore, through this process, we are going to sequence the amino acids from the amino terminus one by one. In this way, the sequence of a given peptide is determined. Now, when proteins are broken down into fragments, so there are many different peptides which are produced. Each of those peptides is sequenced by using the process of Edman degradation. Once all of the peptides have been sequenced of a given protein, now the problem is the assembly, that in what order these peptides were arranged on that polypeptide. This is done by using the following scheme. The given protein which is to be sequenced is fragmented into its constituent peptides in two separate reaction mixtures by two different reagents. In this particular example, you can see the sequence of a peptide here, which is shown in the middle of the picture. In two different chemical reactions, two different types of substances are used for the breakdown of this peptide. One such compound which is used is a chemical substance which is called as cyanogen bromide, CNBr. Cyanogen bromide, it breaks down the peptide bond after a methionine. So wherever it encounters a methionine, it will break down the peptide bond. Here, here, to produce these given three fragments. So this is one reaction mixture. Now the fragments which are produced by degradation by cyanogen bromide, these fragments are purified and then eventually sequenced. The order of these peptides is not known to us. In another reaction mixture, the protein to be sequenced is treated with trypsin. Trypsin hydrolyzes the peptide bond after a positively charged amino acid. So lysine and arginine, these are positively charged amino acids. So therefore, upon hydrolysis by trypsin, these three fragments will be produced as represented by these arrows. And after hydrolysis, each of the fragment produced is sequenced through Edman degradation. Again, the order of these fragments is not known to us. So there are two different reaction mixtures, and in each reaction mixture, different type of hydrolyzing substance is used to fragment the protein into peptides. After this, the sequence is determined. So over here, you can see that these are the fragments 1, 2, and 3, which are produced by treatment with cyanogen bromide. And these are the fragments produced by trypsin. Again, these are three fragments which are obtained after hydrolysis by trypsin. So but we do not know in what order these three peptides are arranged. Similarly, we do not know that in what order these three peptides were arranged in this reaction mixture. So the way we reconstruct the sequence is to compare the sequence of amino acids between these two series of fragments. We can randomly start from any of these sequences. So let us first look at this peptide: aspartic acid, glycine, arginine, cysteine, alanine, glutamine. So now we try to look for matches of sequence of this peptide with the other peptides obtained by the hydrolysis by trypsin. So for example, you see that this is cysteine, alanine, glutamine. So in this fragment, we find the same sequence: cysteine, alanine, glutamine. So we can just align it here. Then the sequence is aspartate, glycine, arginine. So we can look at the sequence. Here you can see aspartate, glycine, arginine, the same sequence here. So that means that this fragment is going to align here. So we put this fragment here, and now we have leucine, proline, methionine from the tryptic digest. So we trace these amino acids in cyanogen bromide digested fragments. So now we look at leucine, proline, and methionine. There it is. So you see leucine, proline, methionine. So that means this fragment is going to align here. And now we find another portion here: glycine, alanine, lysine. So we look for, so there you go, glycine, alanine, and lysine. So that means this fragment is going to align here. And then we have phenylalanine, tryptophan, methionine. So that means this portion, this fragment is going to align here. So in this way, the process of breakdown or fragmentation of a protein is done in two different reaction mixtures by using two different types of hydrolyzing agents. Then, the fragments produced in one case are separated and sequenced. The fragments produced in the second case are also purified and sequenced, and then we find the overlaps between these fragments which are produced by hydrolysis in these two reaction mixtures. By comparing these sequences and finding the overlaps, we can reconstruct the sequence of the original protein. So in this example, you can see that the sequence of this protein will be phenylalanine, tryptophan, methionine, glycine, alanine, lysine, leucine, proline, methionine, aspartate, glycine, arginine, cysteine, alanine, and glutamine. So that will be the finally reconstructed sequence of this protein. So this is the experimental method to determine the primary structure of the protein. This is a short sequence of amino acids, so we can do it manually. And also we have only two types of hydrolyzing agents. So therefore this could be conveniently done in a manual fashion. But when this has to be done for larger proteins or for more than two hydrolyzing agents, we have got computational programs which automatically find these overlaps and they give us the assembled sequence automatically. So this is an end of chapter problem from Voigt and Voigt, which shows the sequence of fragments obtained by hydrolysis through cyanogen bromide and trypsin. So this will be a practice problem for you to compare the sequences, find overlaps, and assemble the sequence of the original peptide. If you find any difficulty in this question, you can surely write it to me. Now, quickly, I'm going to talk about the various methods of purification for proteins. These include chromatography as well as electrophoresis. Admin degradation is a method which uses a large amount of protein. So sufficient amount of protein has to be purified from a given source. And after purification, then the protein is subjected to hydrolysis and fragmentation. So here we are going to quickly look at these two important methods of purification of proteins. The first one is chromatography. Chromatography is a separation method in which the molecules are separated on the basis of their relative binding affinity. there is a stationary phase and a mobile phase. As the name says, the mobile phase flows through the stationary phase. The substances which are to be separated here, which are going to be proteins. So proteins which are to be separated, if they have stronger binding affinity with the stationary phase, they will bind to that stationary phase and they will stay in that column. However, if the proteins they have high binding affinity to the mobile phase, they will have little interaction with the stationary phase, they will flow out through that column. So depending upon the various types of properties, we can separate proteins. Proteins could be separated on the basis of their size, charge, hydrophobicity, or other specific interactions. So the first one represented here is called as ion exchange chromatography. This is called as ion exchange because the stationary phase, which is represented by these small blue circles, the stationary phase contains charges on it. So here you can see in this magnified view that these are the polymer beads which contain negatively charged functional groups on them. So the beads are going to be negatively charged. This is the protein solution which is then added to the stationary phase and it moves through the stationary phase. Those proteins which are negatively charged, they will be repelled by the stationary phase because the stationary phase is also negative. So the negative charged proteins will be repelled by the stationary phase and they will move through the column and will eventually come out of the column. The process of removing a protein when a protein it leaves the column, that is called as elution. So these proteins which are negatively charged, they will have repulsive forces with the column, they will not bind to the stationary phase and they will be immediately eluted from the column. Those proteins which have positive charges, they will bind to the column as shown here. Those proteins which have a little positive charge, their binding will be weaker. Those proteins which have high positive charge on them, their binding affinity with the column will be stronger. Now, once the proteins are loaded onto this column and they are bound to the column, now the proteins, they have to be released from the column. This is done by putting in competing ions. The column is negatively charged, so we put in competing positive ions. Mostly, those are the ions of salt. Sodium chloride is the commonly used salt. So when we add a solution of sodium chloride with increasing concentration of sodium ions, these ions are going to replace the already bound proteins from the columns. Those proteins which have weak binding with the column, they will be released at low concentration of sodium ions. And therefore, they will be eluted from the column, they could be collected in the collection tubes. Then we increase the sodium concentration in the solution. And as sodium concentration increase, now the proteins which have relatively high affinity with the column, they will be displaced by sodium ions. So sodium ions are going to replace them and occupy their position and release those proteins from the column. And now the other proteins, they will be released from the column. Those proteins which have strongest binding affinity, they will need even higher concentration of sodium ions to be released from the column. So as we change the salt concentration in this buffer, we collect the eluted proteins in different tubes, in different collection tubes. So the proteins are going to be separated from one another based upon their charges. So in this type of chromatography, the stationary phase has a certain charge, which is negative in this case. Positively charged ions, which are the proteins, are going to come and bind here. And later, those proteins are going to be exchanged by the sodium ions. When there is an excess of sodium ions, they're going to replace the proteins and release the proteins from the stationary phase. Since ions are going to be exchanged on the stationary phase, so this type of chromatography is called as ion exchange chromatography. Here, positive charges are going to be exchanged. So this is cation exchange chromatography. If the stationary phase contains positive charges, then negative charges are going to be exchanged on the stationary phase. That is called as anion exchange chromatography. So ion exchange chromatography is one important method of separation of proteins, and this is based upon the charge on given proteins. The charge on proteins is dependent upon pH. So by changing the pH, the same protein can have different magnitude of charge on it. The next type of chromatography is called as gel filtration chromatography. It is sometimes also called as size exclusion chromatography. This type of chromatography separates the molecules on the basis of their molecular mass. This is the stationary phase shown by means of these circles, and this is a magnified view of the beads of the stationary phase. The beads are porous, they contain pores in them. The pores or the channels have a certain size limit. Those protein molecules which are smaller, they can go inside, they can move inside these channels which are present in the beads. Those protein molecules which are larger, they are excluded. They will not enter the bead and they will remain outside the beads. So now we add the solution of proteins containing several proteins in it with different sizes. As these proteins they migrate through the column, the larger proteins which are unable to enter the beads of the channels of the beads, they will move through the interstitial spaces. They will move through the spaces between the beads and they will move at a rapid rate. Those proteins which are smaller, as shown here, they have smaller dimensions as compared to the size of the channels in the bead. So these proteins they will penetrate through the beads. They will move through these channels in the bead. And when they exit one bead, now they can enter another bead. And then through this, a third bead, a fourth bead, and so on. So these proteins, which are smaller in size, and they can enter through the beads, enter through these channels, they are going to move through all these various channels present in the beads. And hence, they are going to take longer. They will travel greater distance to reach the end of the chromatography column. Large proteins, on the other hand, which do not enter these channels, they will move through the interstitial spaces and they will be eluted first. So gel filtration chromatography separates proteins on the basis of their molecular size and shape. Larger proteins are going to be eluted first. They will be collected in a collection tube. Smaller proteins will take longer to be eluted from the column and hence could be collected in a different collection tube. In this way, proteins could be separated from one another based upon their molecular size. This process is called as gel filtration chromatography. The next type of chromatography is called as affinity chromatography. This chromatography is based upon specific interaction of a protein with some ligand. So on the stationary phase, the ligand for a given protein is attached. This is shown by means of these black circles. So these are the ligand molecules which bind specifically to this protein of interest. So these proteins which are crescent shaped, only they are going to bind to these ligands. Other proteins, they are unable to interact with this, so therefore they will not bind to the stationary phase. The ligand is covalently attached to the stationary phase. So now the mixture of the proteins is added. Only these red colored protein molecules which are specific for this ligand, they will bind to the stationary phase. All other proteins, since they are unable to bind to this ligand, so they will be eluted from the column. Later, we put an excess of the ligand solution which is going to displace the protein of interest from the column, and it could be collected in separate collection tubes. So here, the separation is based upon specific interaction of a protein with its binding ligand. So this is based upon specific binding affinity. For example, if an enzyme has to be separated, then the substrate for that enzyme could be coated onto the stationary phase, and the bound substrate is going to capture the enzyme of interest, and it will not bind the other proteins present in the given mixture. So in this way, the protein, based upon its specific binding affinity to its ligand, is captured on the stationary phase and hence is purified. Since this type of chromatography is based upon specific binding affinity, so this is called as affinity chromatography. The next type of separation method is electrophoresis. Electrophoresis is a separation technique in which charged molecules are subjected to an electric field. Proteins are also charged molecules. So these charged molecules, in this particular case, the proteins, when they are put in an electric field, they will migrate depending upon their charge to mass ratio. This picture shows a diagrammatic representation of a type of electrophoresis called as SDS-PAGE. It is an abbreviation for sodium dodecyl sulfate polyacrylamide gel electrophoresis. This gel is a polyacrylamide gel. Polyacrylamide gel is produced by the polymerization of acrylamide and bisacrylamide monomers. After polymerization, the gel is cast, and the gel is cast in the presence of this detergent SDS. SDS denatures the protein and gives negative charge to all of the proteins. So now when proteins are, after casting the gel, they are separated through this gel, the proteins are going to migrate based upon their molecular mass. The proteins which are smaller in size, they will migrate faster. The proteins which are larger in size, they will migrate slower. So the proteins which are smaller, they will travel greater distance. As shown in this figure, you can see the bands of many different bands of proteins. So the proteins which are smaller in size, they migrate faster and they are going to travel more distance, as compared to the proteins which are larger in size and hence travel less distance. So this method is not only used for separation of the proteins, but this is also very useful in determining the molecular mass of a given protein. This is done in the following manner. This is a protein of interest whose mass is to be determined. This protein solution was loaded into well number 2 of the gel. The well number 1 of the gel contains a mixture of proteins with known molecular mass. This is called as a ladder, or sometimes called as the molecular mass marker. The marker contains, it is a mixture of proteins and the mass of each of the protein is known to us. Since separation through SDS-PAGE is based upon the molecular mass, so the rate of migration are proportional to the molecular mass of given protein. So here you can see that this protein is of 45,000 Dalton and this protein is of 31,000 Dalton. Dalton is a unit used to describe the mass of biological molecules. 1 Dalton is similar to 1 atomic mass unit. In other words, this protein is a 45 kilodalton. The word kilo means 1,000. So this is a 45 kilodalton protein. This one is a 31 kilodalton protein. The protein of interest is residing somewhere in between these two proteins. So by using this comparison, we can deduce that the protein of interest has a molecular mass between 45 kilodalton and 31 kilodalton. We can do a visual estimation through this gel, and we can precisely determine the molecular mass of this given protein by using image analysis softwares. The image analysis softwares, they do a manipulation which is diagrammatically represented here. On the x-axis is the migration, the rate of migration, and on the y-axis is the log of the molecular mass. So these dots are representing the standard proteins whose molecular mass are known, and this is the migration rate of the protein whose mass is to be determined. So we calculate the migration rate and we trace it back onto this graph to find the molecular mass of a given protein. So these days we have got sophisticated computational softwares which can do this work for us. So we do not need to do this manually, and this could be automatically determined by those computer softwares. So one important significance of electrophoresis is that it is not only a tool to separate and purify proteins, but also it is used for determination of the molecular mass and the concentration of proteins. The concentration of a given protein can also be determined by using SDS-PAGE, but right now we are not going to talk about that. In the next session, we will talk more about the secondary structure of proteins.